welcome to today's deep dive. If you're tuning in, you probably have, this kind of insatiable curiosity about how things actually work.
Especially the really fundamental mechanics of your own body.
Right, exactly.
Yeah.
We love exploring those mysteries with you. So to start us off today, I want you to just think about what you were doing right this second.
Probably sitting down or maybe driving.
Yeah, sitting on a couch or commuting on a train, feeling completely at rest. But inside your body, it is absolute thermodynamic chaos.
Oh, complete chaos.
Right. There's this relentless, microscopic battle happening in almost every single one of your cells. And if it were to pause for, even just a few minutes, the consequences would be catastrophic.
It's a really profound illusion, actually, that whole feeling of being at rest.
Yeah.
I mean, we tend to think of the human body like a parked car. You turn the engine off, you put the keys away, and the machine just sits there.
Just totally inert, not burning fuel.
Exactly. But biologically, down at the cellular level, maintaining a state of quote-unquote rest requires an astonishing amount of continuous, grueling work.
And that brings us to the mission for today's deep dive. We are exploring the cell's essential engine, the sodium-potassium pump.
Formerly known as the NAS, ATPase.
Right. And we're going to unpack how this tiny, invisible machine maintains the chemical gradients that make literally every nerve impulse and, every muscle contraction in your body possible.
It is, without exaggeration, one of the most important biological mechanisms ever discovered.
I mean, it powers every heartbeat, right?
Every heartbeat, every muscle flex, and every single thought you are having right now as you process the words we're saying.
I want to put a specific number on exactly how hard this machine works, because there's a statistic in our sources that completely rewired how I view my own biology.
The energy expenditure one.
Yes. Roughly a fifth to a quarter of your entire resting metabolic energy is spent running this single enzyme.
Just this one microscopic pump.
Just one.
Yeah.
And if you look at the brain, the majority of the energy your brain consumes is dedicated just to powering these pumps.
The scale of that is just staggering. I mean, your brain accounts for, maybe 2% of your total body weight?
Right, it's tiny.
But it hoovers up this massive, disproportionate amount of your oxygen and glucose. And it's doing that largely just to pay the energy bill for this specific mechanism.
So the natural question we have to start with is, what on earth is this pump doing that requires 25% of our daily fuel budget?
Well, it requires all that fuel because the cell is fighting a constant uphill battle against physics.
Okay, unpack that a bit.
Every cell in your body needs to maintain a very strict asymmetry in its ion concentrations just to function. So, to put it simply, you need a really high concentration of sodium outside the cell and very little inside.
Okay.
And conversely, you need a high concentration of potassium inside the cell and very little outside.
And it's a pretty steep difference, right?
Very. It's roughly a 10-fold gradient for sodium and a 30-fold gradient for potassium.
And nature absolutely hates a gradient.
Right. Left to their own devices, diffusion would just cause those ions to flow down their gradients until everything was perfectly equalized inside and out.
The cell would lose all that asymmetry.
Exactly. The sodium-potassium pump is the molecular machine that is constantly pushing those ions back against the tide to maintain that non-equilibrium state.
I want to visualize the actual mechanism here, how it does this heavy lifting. Because going through the research, it's not a fair trade. It's a very specific exchange.
It operates on a strict 3-to-2 stoichiometry.
Stoichiometry just being the technical term for the chemical ratio.
Precisely. It's highly specific. So for every one molecule of ATP, it burns.
ATP being the basic energy currency for cells.
Right. For every one ATP, the pump physically changes its shape. It grabs 3 sodium ions from inside the cell and shoves them out.
Okay, 3 out.
And then it grabs 2 potassium ions from outside the cell and pulls them in.
And it's shoving these ions against their natural concentration gradients.
Exactly. That physical resistance is exactly why it requires so much ATP.
Okay, so reading through the history of how early physiologists viewed this, I actually came up with an analogy. I want to run this leaky boat idea by you and see if it holds water.
Let's hear it.
So back in the early 20th century, around 1902, scientists like Julius Bernstein thought the cell's resting state was a passive equilibrium.
Right, the old classical view.
Yeah. And I picture this like a perfectly watertight boat sitting peacefully on a calm, glassy lake. It's balanced, it's not moving, it doesn't need to burn any fuel to stay afloat.
Sounds relaxing.
Right. But based on what you're saying, that is entirely wrong. The cell is actually a leaky boat.
Very leaky.
It's constantly taking on water, which would be the sodium leaking in, and it's constantly losing its buoyant air, the potassium leaking out.
Okay, I follow.
So the sodium-potassium pump is essentially an exhausted sailor in the bottom of that boat, just furiously bailing water out and pumping air back in 24 hours a day just to keep the boat from sinking into the lake.
I love that visualization because it really captures the sheer desperation of the cell's baseline state.
Sounds stressful.
It is. The cell membrane is never perfectly impermeable. There are always these passive channels open, allowing ions to leak.
Right.
So if the sailor stops bailing, those chemical gradients collapse within minutes. And if those gradients collapse, the cell can no longer fire an action potential.
Which is the electrical signal nerves use to communicate.
Exactly. The neuron becomes inert, it dies. This realization completely shifted how we understand biology. The resting state is not an equilibrium at all. It's a steady state, actively maintained against constant leak.
Okay, but this brings up a massive misconception that I totally fell for when I first started looking into this.
Oh, what was that?
Well, I read that the pump pushes out 3 positive charges, the 3 sodiums, and only takes in 2 positive charges, the potassiums.
Right.
So that means there is a net loss of 1 positive charge from inside the cell every single cycle.
Yes.
My immediate thought was, oh, this pump creates the cell's electrical charge. This is the battery.
You are definitely not the only one to make that assumption. It's incredibly common to think the pump itself is the battery.
No, it's not.
Well, what you're describing is the fact that the pump is electrogenic, meaning it does move an unequal electrical charge across the membrane.
Right.
But here's the crucial correction. That direct pumping action only accounts for a tiny, tiny fraction of the cell's resting membrane potential.
Really? Like how much?
We're talking maybe 2 to 10 millivolts out of a total negative 70 millivolts in a typical neuron.
Which is wild to me. Because if the pump isn't generating the bulk of that negative 70 millivolt charge, where is all that electricity actually coming from?
It comes from the potassium leaking back out.
Wait, unpack that for me.
Okay, so the pump's real job isn't to create the electrical charge directly. Its job is to build the concentration gradient. It packs a massive amount of potassium inside the cell.
Like blowing up a balloon.
Right, and because it's so crowded in there, that potassium naturally wants to diffuse back out through those passive leak channels we just talked about.
Okay, the leak in the boat.
Exactly. Now, as those positively charged potassium ions leave the cell to follow their chemical gradient, they leave behind these large, negatively charged proteins and molecules inside the cell.
And those are too big to cross the membrane.
Yes, they're simply too big to escape.
Ah, so the positive charge leaves, and the negative charge is just trapped inside.
Yes. That separation of charge, caused entirely by the passive leak of potassium, is what actually generates the vast majority of the negative voltage inside the cell.
Oh.
The pump builds the gradient, but the leak channels create the voltage.
So to go back to my exhausted sailor, he bails the water out, specifically so the water can leak back in and turn like a water wheel that powers the ship.
That is a fantastic way to extend the analogy. The leak does the actual electrical work.
So how did anyone figure this out? I mean, this sailor is microscopic. We can't just look at a human cell under a microscope and see a tiny protein-bailing sodium.
No, you definitely can't.
The journey to proving this invisible machine existed is just fascinating because researchers had to look at some extreme examples in the animal kingdom just to get a glimpse of it.
They did. We have to rewind to the early 1950s, looking at the work of scientists Alan Hodgkin and Richard Keynes. They were trying to prove that ions were actively being moved.
Right.
And to do it, they used the squid giant axon.
Just to clarify for everyone, when biologists say giant axon, they aren't talking about like a giant mythical squid, right?
Yeah.
They mean the nerve fiber itself is physically huge.
Yes, exactly. In some squid, a single nerve fiber controlling their propulsion mantle can be up to 1 millimeter thick.
Wow.
That is massive for a single cell. It's thick enough that you can actually insert tiny wires and pipettes right into it relatively easily in a laboratory setting.
That makes total sense as to why it's such a classic model organism.
Right. So Hodgkin and Keynes used a biological tracer. They used radioactive sodium, specifically the isotope sodium-24. They loaded this radioactive sodium into the giant nerve cell, and then they essentially set up a Geiger counter nearby. And they watched, or rather listened, as the radioactive sodium was continuously pushed out of the cell.
Which proves the sodium is moving out, but it doesn't necessarily prove a specialized machine is burning fuel to do the work, does it?
No, it doesn't. And that is where the genius of their experiment comes in. While the sodium was being pumped out, they suddenly poisoned the cell's metabolism using chemicals like dinitrophenol or cyanide.
Cyanide halts ATP production, right? It suffocates the cell's energy supply.
Correct. And the moment the cell ran out of ATP, the radioactive sodium extrusion stopped dead.
Even though the gradient was still there? I mean, the sodium still, quote-unquote, wanted to move, but it couldn't.
It couldn't move. This proved, fundamentally, that the extrusion required active thermodynamic work. It required an energy source.
Wow.
This was the first principles proof of a sodium pump, years before anyone had actually isolated the physical molecule doing the pumping.
Which perfectly sets the stage for how we found the physical pump itself. Enter Jens Christian Skau, a Danish physician and biophysicist in 1957.
Yes.
And what I love about this story is the serendipity of it. Skau was not trying to find the sodium pump. He wasn't even working on basic cellular resting states.
No, he was investigating how local anesthetics worked. He wanted to understand how certain drugs numbed nerves. And his animal model of choice wasn't the squid, it was the shore crab.
What, crabs?
Well, if you want to study nerve membranes, shore crabs have these large leg nerves that are incredibly dense with membrane tissue. You can harvest a large amount of it relatively quickly.
So he's essentially grinding up crab leg nerves in the lab looking for how anesthetics bind.
More or less. And in doing so, he isolated an enzyme in these crab nerve membranes that splits ATP. It was an ATPase, an enzyme that consumes energy.
Yeah.
But he noticed something incredibly perplexing about its behavior. The enzyme's activity was very sluggish, almost dormant, until he added sodium, potassium, and magnesium to the solution all at the exact same time.
Meaning if he just added sodium, nothing happened.
Very little happened. Neither sodium alone nor potassium alone activated it to any significant degree. It demanded all of them present.
And that codependence is the absolute fingerprint of the machine.
Skau realized the profound implication of that. If the enzyme only fires and burns ATP when both sodium and potassium are present, it must be a transporter that requires both ions simultaneously to do its job.
That's amazing.
He had found the biochemical engine that Hodgkin and Keynes had predicted indirectly with their radioactive squid nerves.
And he published this monumental finding in 1957. But here is a detail that really highlights the slow grind of scientific consensus. Skau didn't win the Nobel Prize in chemistry for this discovery until 1997.
Yeah.
It took 40 years. It is a stark reminder of how long it takes for foundational biochemistry to be fully recognized and, integrated into our broader understanding of human life.
It truly is a long arc. But once that foundation was firmly laid, it revolutionized our understanding of human health.
Yeah.
If we transition from the history of the lab bench to the high stakes of the emergency room, understanding this pump isn't just an exercise in textbook trivia. It dictates the literal line between life and death when a human body begins to fail.
I wanted to explore what happens when that exhausted sailor we talked about earlier is completely starved of food. Let's look at a stroke.
A very grim but important example.
Specifically an ischemic stroke, where a blood clot blocks blood flow to a part of the brain. There's this common phrase emergency doctors use, time is brain. And reading the sources, that phrase is a direct consequence of the sodium-potassium pump failing.
Right. Time is brain is a literal description of cellular thermodynamics unraveling. When a clot blocks blood flow, the delivery of oxygen and glucose to that region of the brain instantly stops.
And without oxygen and glucose, the neurons can't produce ATP.
The fuel supply collapses within minutes.
And without ATP, the sailor stops bailing.
The pump stops running entirely. Almost immediately, you experience what neurologists call anoxic depolarization. Because the pump isn't pushing ions back, the gradients collapse.
So sodium and calcium, which are highly concentrated outside the cell, just flood in.
Yes. And the potassium leaks out.
The carefully maintained asymmetry just washes away.
Completely. And when that happens, basic physics takes over. Where sodium goes, water follows through osmosis.
Oh, no.
So water rapidly rushes into the neuron, causing it to swell up massively. In medicine, this is called cytotoxic edema.
Cyto meaning cell, toxic meaning deadly, and edema meaning swelling.
Exactly. The neuron literally swells until it's critically damaged. But as bad as the swelling is, the influx of calcium is far more destructive.
How does calcium make it worse? I mean, swelling sounds pretty bad on its own.
Well, inside a neuron, calcium acts as a molecular trigger. When a massive wave of calcium floods the cell, it forces the neuron to indiscriminately dump all of its stored neurotransmitters, primarily one called glutamate.
And glutamate is a messenger that tells other neurons to fire, right?
Yes, it's an excitatory neurotransmitter, but in this scenario, it's an uncontrolled torrential flood of glutamate. It washes over all the neighboring healthy neurons, just screaming at them to fire endlessly.
God.
Those neighboring neurons panic. They fire over and over, rapidly depleting their own ATP reserves, trying to reset themselves.
Which causes their own pumps to fail.
Right, which allows calcium to flood into them, releasing more glutamate.
It's a microscopic domino effect.
It is a deadly, expanding wave of cell death known as excitotoxicity. The brain cells essentially excite themselves to death.
That is horrifying.
The damage spreads well beyond the original site of the blocked blood flow. And this entire catastrophic cascade, the loss of motor function, the loss of speech, it all starts because one specific microscopic pump ran out of fuel.
That is exactly why restoring blood flow so rapidly in a stroke is so critical. You are literally racing to feed the sailor before the boat sinks.
Exactly.
That is harrowing to visualize. But knowing how deadly it is when the pump stops makes the next medical application we found genuinely shocking to me. Sometimes we treat patients by intentionally poisoning this very pump.
We do, in highly controlled circumstances.
Right, we're talking about heart failure and a class of drugs called cardiac glycosides, like digoxin,
Yes, digoxin.
which is a purified compound derived from the foxglove plant. And from what I understand, it works by binding directly to the outside of the sodium-potassium pump in heart muscle cells and partially inhibiting it. It slows the sailor down.
That is the exact mechanism of action.
I have to pause on this. We treat a failing, weakened heart by deliberately poisoning the very enzyme that keeps the heart cells alive. That feels incredibly counterintuitive.
I know it does.
It's like slightly severing a car's brake lines to make the engine run hotter. How does intentionally weakening the cell help a failing heart?
Your analogy of cutting the brakes is remarkably apt, actually, because it initiates a very dangerous game of physiological dominoes. Let's walk through the mechanics.
Okay, lay it on me.
When you administer digoxin, it slows down the sodium-potassium pump. Because the pump is working slower, the sodium concentration inside the heart cell rises just a little bit. The gradient gets a little weaker.
Okay, I follow that. Less bailing means a bit more water in the boat.
Now, there is another transporter embedded in the heart cell's membrane called the sodium-calcium exchanger, or NCX.
NCX, okay.
The NCX is essentially a molecular turnstile. Normally, it uses the strong inward rush of sodium, the gradient the pump built as energy, to pump calcium out of the cell. It relies on the steepness of that sodium gradient to function.
So it's piggybacking on the pump's hard work.
Yes. But because the digoxin weakened the sodium gradient, the NCX turnstile slows down. It loses its driving force, so it doesn't pump as much calcium out, which means more calcium lingers inside the heart muscle cell.
Wait, and as we discussed earlier, calcium is a molecular trigger. In muscle cells, calcium is what drives the physical contraction.
Precisely. With slightly more calcium lingering inside the cell during every heartbeat, the heart muscle physically contracts more forcefully. In cardiology, this is called a positive inotropic effect. For a patient whose heart is failing, enlarged, and pumping weakly, this drug artificially forces the muscle fibers to squeeze harder, improving blood flow to the rest of the body.
That is incredibly clever biochemistry. Leveraging one pump's gradient to trap calcium using a second pump. But it sounds terrifyingly risky. If you cut the brakes a little too much, you crash the car.
Which is why digoxin has an infamously narrow therapeutic window. It is a staple teaching case in medical pharmacology.
Because the line between helping and hurting is so thin.
Exactly. Give a patient slightly too little of the drug, and it does absolutely nothing. Give them slightly too much, and you poison the pump so severely that the heart cell's electrical gradients collapse entirely.
Which would cause arrhythmias.
Severe, life-threatening arrhythmias. Doctors have to monitor digoxin blood levels incredibly closely because everyday factors like poor kidney function or even just low dietary potassium can suddenly shift a perfectly safe dose into a toxic, lethal dose.
That's wild. Now, if this mechanism is so incredibly delicate and manipulating it is so dangerous, it makes me wonder about things outside the controlled environment of a pharmacy. Because digoxin is a purified drug, but these compounds exist in nature.
They do.
Like ubain, which our sources note was traditionally used as an East African arrow poison.
Yes, uabain is a structurally similar plant compound, and it is a very potent, irreversible inhibitor of the pump. If it enters the bloodstream, it stops the heart.
Because these natural plant toxins have such a profound, specific effect on our most vital cellular engine, It led to some intense historical debates in biology, didn't it?
Oh, it sparked a massive scientific hunt in the late 20th century. Researchers started asking a very logical question. Why do human cells have a highly specific binding site, a receptor for an obscure African plant poison and a European flower?
Right. Nature wouldn't evolve a very specific keyhole on our cells unless the human body made its own internal key to fit it.
That was the driving hypothesis. They hunted for what they called endogenous digitalis-like factors.
Endogenous meaning internally produced.
Right. The compelling idea was that the human body must produce its own internal microscopic version of uabane, perhaps a hormone secreted by the adrenal glands or the brain.
And we use that to naturally regulate our own blood pressure by tweaking the activity of the pump.
That was the
It
theory.
makes perfect sense. So did they find the internal key?
Well, it became one of the most fiercely debated topics in physiology. Some research groups published papers claiming they had successfully isolated these endogenous uabane compounds circulating in human plasma,
Right.
particularly in patients suffering from severely high blood pressure. They argued it was a major missing link in hypertension.
But I sense a however coming.
However, other prominent labs could not replicate those findings at all. They strongly questioned the validity of the chemical assays being used, suggesting they were picking up false positives.
The classic replication crisis.
Exactly. It remains a highly contentious topic today. While the idea is beautiful in theory, the broader scientific field is much more cautious and skeptical now than those early enthusiastic claims suggested.
I find it fascinating how a microscopic pump can spark such massive, career-defining medical debates. And while we're on the topic of the side effects of manipulating this pump, there is a famous medical mystery tied to digoxin toxicity that I just have to bring up.
I think I know where this is going.
We mentioned that taking too much foxglove extract poisons the pump. One of the bizarre symptoms of that toxicity is visual disturbances. Specifically, patients report seeing colored halos around lights, and their overall vision gets tinted yellow-green.
Yes, the clinical term for that is xanthopsia. Poisoning the pump alters the delicate ion gradients in the photoreceptor cells of the retina, which changes how the eye processes color and light.
So there is this incredibly famous theory in art history, the Vincent van Gogh's Yellow Period, where his late brilliant paintings were suddenly dominated by these intense swirling yellows and massive halos around the stars, was actually caused by digitalis toxicity.
Yes, the Van Gogh theory.
Because we know he was being treated by a physician, Dr. Gachet, who commonly prescribed foxglove extracts. He even painted Dr. Gachet holding a foxglove plant. It's right there on the canvas.
It is an incredibly vivid, compelling story. It really is. And it's beloved in medical schools. Professors use it all the time as a mnemonic device to help students remember the visual symptoms of digoxin toxicity.
I love it when art history and cellular biochemistry collide to solve a mystery like that. It's so satisfying.
Well, as a guide to the actual research, I unfortunately have to be the voice of reality here and rein you in slightly.
Oh no, don't ruin it for me.
I have to. While it is a beautiful narrative that perfectly connects the dots, medical historians and physicians highly dispute the evidence for it.
Really, but it fits so well.
It fits almost too well. The reality is that Van Gogh was treated with many different compounds, including absinthe and bromide. He suffered from complex, overlapping mental and physical illnesses.
Right, he had a lot going on.
And creatively, he was deeply influenced by the color theories of the time and the vibrant light of southern France. There are many alternative, highly plausible explanations for his artistic choices.
So they're more of a fun myth.
It is a fascinating, contested theory, not established historical fact. We always have to keep our critical thinking intact when a diagnostic story from 100 years ago sounds just a little too perfect.
Rare enough. The reality check is appreciated. I guess we will leave Van Gogh to the art historians. But even without that connection, the sheer scope of what this single pump controls is just unbelievable.
It really is.
Let's tie these threads together. What we've seen today is that the sodium-potassium pump isn't just an obscure metabolic detail you memorize to pass a biology test. With its precise 3-to-2 electrogenic exchange, it is the literal load-bearing pillar of all excitable tissue.
It is the foundation. It powers every breath you take, every physical movement you make, and your fundamental ability to comprehend the world around you.
Which brings us to a final mind-expanding thought from our sources that I want you, the listener, to ponder as we wrap up today's deep dive. We've talked extensively about what happens when the pump is starved of energy during a stroke, or intentionally poisoned by drugs or plants.
Right.
But what happens if the genetic blueprint for the machine itself is just slightly miswritten from the very beginning?
Ah, you were referring to the rare genetic mutations found in the pump's specific subunit genes.
Yes. Researchers have identified mutations in a gene called ATP1A2. And these mutations are directly linked to familial hemiplegic migraine, which is this incredibly severe type of migraine that causes temporary paralysis on one side of the body.
Right.
And mutations in another gene, ATP1A3, are linked to rapid onset dystonia Parkinsonism, which is a profound, debilitating movement disorder.
It is a profound realization to end on, honestly, because when we study those conditions, we are witnessing the systemic, body-wide effects of a subtle structural miswiring of just this one microscopic machine from the moment of birth.
Just one tiny error in the blueprint.
Exactly. It alters how neurons reset, which fundamentally alters human consciousness, pain perception, and physical autonomy.
It is a beautiful, if fragile, reminder of how life stays functional from moment to moment. I mean, we are entirely unknowingly dependent on that exhausted sailor, constantly bailing water out of the leaky boat, never allowed to rest, just so we can sit here and experience the illusion of resting.
A perfect synthesis. The biological work never stops, and we should be very grateful for that hidden labor.
Thank you so much for joining us on this deep dive into the cell's essential engine. Keep questioning, keep learning, and we will catch you next time.